Hi, my name is Chrissy Judd and I'll be studying the effects of oxybenzone on colour change and background choices in chameleon prawns. So there's two main components to my study, the oxybenzone, which is the chemical, and chameleon prawns, which are my chosen study species. So oxybenzone is a UV blocking chemical and I'm investigating whether it has an effect on the camouflage capabilities of chameleon prawns. So I'll speak a bit more about oxybenzone later, but chameleon prawns are found all over the UK and mostly recorded in the south, such as in Cornwall. They're a small species which exhibit a variety of colour forms that subjectively show a close similarity to seaweed species on which they associate. So research at the end of the 19th century revealed that the species is capable of changing colour over different timescales, including driven by light intensity, periodic shifts between diurnal nocturnal forms, and slow changes in response to novel substrates. Cryptic coloration allowing visual camouflage is an anti-predator strategy in nature and provides a classic example of evolution by natural selection. Crypsis works by reducing the chance of prey detection or recognition by the visual system of potential predators, often through background matching. So, oxybenzone, what is it? Oxybenzone is used in plastics as an ultraviolet light absorber and stabilizer. It is used along with other benzophenones in sunscreens, hairsprays, and cosmetics, and it helps prevent potential damage from sunlight exposure. It is also found in concentrations of up to 1% in nail polishes. Oxybenzone, which is in this little tub, can also be used as a photostabilizer for synthetic resins, and benzophenones can leach from food packages and are widely used as photoinitiators to activate a chemical that dries ink faster. So why do we choose it? Oxybenzone is clearly found everywhere. It's just this little yellow powder. There's very limited research on the effects of oxybenzone on any temperate species at all. It's been studied extensively in the tropics, but not at all in temperate waters. But UV filters have been detected in coastal waters in the UK, and they reach coastal waters either directly from washing off swimmers or indirectly from wastewater treatment plants. Many of these components have also been found in marine biota, including fish, mollusks, and corals, as well as sediments. Where sunscreen components have been detected, concentrations are variable, so they're generally found at barely detectable levels, but much higher concentrations over 1 part per million have been reported. So what do we already know? We know that oxybenzone has contributed to the bleaching of coral fragments and coral cells from different species of hard coral, damage and deformation of coral larvae, and have contributed to the damage of coral DNA and their reproductive success. So the first part of my study was colour change. So I hypothesised that chameleon prawns' circadian colour change will be adversely affected by oxybenzone, and predicted that chameleon prawns will change their blue nighttime morph less frequently under the influence of oxybenzone. We already know that crustaceans, particularly those living in dynamic environments such as rock pools, have frequently been used as key organisms to investigate the fitness advantages conveyed by cryptic traits. Chameleon prawns are a small species that exhibit a variety of colour forms which subjectively show a close similarity to the seaweed species in which they associate. Chameleon prawns undergo day-night changes from their diurnal type to blue-grey at night, and colour change also often occurs over longer periods, so red chameleon prawns change to green one on green seaweed for 20 days, and vice versa. Next part of my experiment was my behavioural trials. So I hypothesised that the background selective behaviour of chameleon prawns will likely be adversely affected by oxybenzone, and predicted that chameleon prawns will choose an incorrect background more frequently under the influence of oxybenzone. So we already know that aside from direct colour adaptations, animals may also facilitate camouflage by behaviourally choosing appropriate backgrounds. This may seem obvious, but there has historically been a lack of consistent experimental investigation into this area, and a few rigorous tests of how and when behavioural choices facilitate crypsis on natural substrates has been conducted. However, recent work has demonstrated the existence of background choice at an individual level for improving concealment. This was done by Sam Green in 2019. However, most studies to date have tested background choices in species where individuals are fixed in appearance, and work has yet to fully consider the importance of behavioural preferences affecting concealment of species capable of colour change. Individuals from species capable of rapid colour alteration might be under less intense selective pressure to rely on behavioural choices for crypsis, since they can rapidly alter appearance regardless of their background. So how did I do this? So my experiment has two parts, the colour change trials and behavioural trials, and all the prawns were collected on Gilly Beach when the tide was lower than a metre. The prawns were collected by dunking a bucket near the seaweed and creating a vacuum, and only red chameleon prawns were used. So in the colour change trials, there were two groups, experimental and control, and in each group there were 30 prawns. The experimental group, everything was done with 250 millilitres of water and 224 micrograms of oxybenzone, and in the control group only 250 millilitres of water. So both groups were placed in seawater only and taken their daytime morph photo, which is their red morph, and then submerged into either their control experimental groups in complete darkness, and then taken a photo of their night morph, which is this like blue-grey or not. The second part of my trial was my behavioural trials, and this was done using a custom-made wide choice decision chamber. So the prawn was put in this bit for a minute to acclimatise either to the chemical or just seawater in this chamber, and then the opaque divider was removed, and there was green seaweed there and red seaweed there, and they were given the choice. They basically had to choose which colour background, and I noted down which one they chose. So my expected results for my colour change, I haven't actually completed the image analysis yet, but in my preliminary results I found that out of the 30 prawns which were tested for colour change under the influence of oxybenzone, only 27% changed their nocturne colour, whereas in the control group 89% changed their nocturne colour. This means that 73% of the prawns in the experimental group did not change to their nocturnal blue-grey morph, compared to only 11% in the control group. And this test was carried out to see whether the oxybenzone would have an effect on circadian colour change in chameleon prawns, and we found that it did. Next part of my experiment was behaviour trials, and this is my preliminary results for that. So on the y-axis, there's the number of prawns, x-axis is experimental trials, and the control trials. The red signifies a red substrate choice, and green signifies a green substrate choice. And as you can see from here, that in the experimental trial, green substrate was chosen significantly more frequently than in the control, and in the control, red substrate was chosen significantly more frequently. Although my data analysis is not complete yet, I did a simple exact binomial test and found that the results from my control group are significant, with a p-value of 0.0002, as well as the results from my experimental group with a p-value of 0.006, with a probability of 50%. And I carried out this test to see whether oxybenzone had an effect on background matching of chameleon prawns, and found again that it does. So what are the implications of this research? So unfortunately, it's almost impossible to discuss whether oxybenzone itself is having an effect on the mechanism of colour change in chameleon prawns, as how the chameleon prawns themselves change this blue-grey morph is unknown, so that would be really interesting and an important area for further research. However, now that we do know that oxybenzone is having an effect, it has major implications for conservation issues, and hopefully could change the way that we look at sunblock in temperate areas as well as in tropical. Furthermore, oxybenzone is a UV filter, and so one of my theories behind why it may be making a difference in the colour change of chameleon prawns is light refraction. If the oxybenzone is changing the way that the light is refracted through water, then this may influence the way that the prawn is perceiving their environment. However, there is no current evidence for this. I believe that this research is important, however, as it's showing the amount of stress anthropogenic factors is putting on animals is completely unknown and is potentially much more harmful than we considered. The effects of sunblock have been studied fairly extensively on coral reefs and tropical symbionts, but essentially not at all in temperate areas. And although you may ask why we specifically care about chameleon prawns, they should be viewed as an ecological model for invertebrates, especially those who use crypsis to survive. So the most important thing about the study is that we have discovered that oxybenzone is affecting anti-predator responses, and further research into this area should be considered. I'd like to thank Martin Stevens and Sam Green for helping with my dissertation, and thank you all very much for listening, and do you have any questions?